Hello, everyone. Thank you very much for joining us in this third talk from the Innova Webinars 2025. This is an event organized by the members of the Innova SPEC project, a transdisciplinary research project focused on innovations in communication in humans and other animals. This is the third of four very interesting talks that will take place over the course of 2 weeks with speakers from different countries on different themes in communication. Please follow our Instagram profile to get more information so you do not miss any of the talks. And two of them are already recorded and available here in the YouTube channel of the Institute of Biosciences. Today, we are very pleased to welcome Dr. Victoria Piai. She's an associate principal investigator at the Donders Institute of Brain, Cognition and Behavior of the Radboud University in the Netherlands. Her research focuses on the neurophysiological basis of language in healthy human adults and individuals with language impairment due to neurological damage. She works with a range of behavioral and neuroimaging methods, particularly electrophysiology, to characterize the neuronal computations required for language and to relate these computations to those employed by memory and cognitive control systems. Additionally, she studies how these computations change following brain injury. Dr. Piai, thank you very much for your presence here today. After your talk, we will have 15 minutes to take questions from the audience. Everyone can ask questions through the YouTube chat. Questions should be in Portuguese. Our team will select and send them to me. Vocês podem perguntar em português e a gente traduz. And Dr. Piai also understands Portuguese. She's Brazilian, so you can ask in Portuguese. Dr. Piai é brasileira, então vocês podem perguntar em português, que ela entende. Qualquer dúvida a gente traduz. This event will be recorded and will be available through YouTube after the transmission. With no more details, I'll give the word to Dr. Piai. Dr. Piai will be here in the background in case of any technical problems.
Let's hope we don't have any. Muitíssimo obrigada pelo convite. Thank you very much for the opportunity to speak to you. It's very exciting. And also thanks so much for having the organization so well done. I'm going to talk about some of my work focusing on language production and in a way a bit of my frustration with not the field as a whole, but at least pockets of the field. So it's kind of a way to try to address that. I looked a little bit into the questions that you as a group are focusing on. And I took a quote from the abstract of that grant. The very interesting, a big question actually, whether there are evolutionary precursors that could have led to the development of cognitive capacities that gave rise to the emergence of, say, the language faculty, amongst other things they were interested in. And I think it's in there that my research interests and my research line very nicely aligns with your overarching goal, in that I'm also interested in the cognitive capacities that underlie language. And so maybe not so much giving rise to it, because I don't have the expertise to look into evolution, but I'm interested in the cognitive capacities that language makes use of. And then, of course, if we talk about the intricate language faculty, we need to actually take a smaller bit of that, because language as a whole is too big of a thing to address. So I focus for this talk, but in general in my research line, on language production, or speech production as many name it. And this, I think, brings us immediately also to what is called the operationalization problem. So here put beautifully into words by a colleague of mine, the problem of justifying the indicative relationship between a behavioral task and a cognitive capacity. So unfortunately, in the type of work, or fortunately, depends on how pessimistic you are, in our way of doing research, we need most of the time to give people tasks that will then tap into the cognitive capacities, the constructs we are interested in. And when we talk about speech production, we immediately get this issue that there's a multitude of tasks to test or to investigate language production or speech production. So just to name a few: picture naming, reading words aloud, word repetition, syllable repetition, and so on. And what you perhaps can see from this already is the fact that some of these tasks will rely on you to retrieve something from memory, something that is more abstract, right? So if you have to name a picture, you actually need to first identify what that concept is, get to the label of that concept, and so on. Whereas for other tasks, say syllable repetition, if I'm doing ba, 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 there's not much memory in there as there's more motor activity. Of course, there's memory for, for movement in itself, but it's not really the same type of memory that I would need for retrieving, identifying a concept, retrieving the word for it, etc. So already in the term speech production, we see that we are, if we are not careful, we could be referring to various different things, depending on the task we are using. And what I focus on, which I want to make very clear, is what I'm calling conceptually driven production, or what is called in the field conceptually driven production. And this means we need to go via the concept to the label of that concept, to the sounds of that concept, right? So there's nothing, I hope you can see my cursor. There's nothing in this image here of a bed that will tell you that this is called bed in English, right? Or cama in Portuguese. There's nothing in the imaging there. So you need to identify that as the concept and then retrieve the label for that concept. And I think some of these issues of, what do we actually mean when we talk about speech production is, you can clearly see that in the literature. And here I'm taking an example, and it's in no way to criticize Jonathan Peele, but I think it nicely illustrates that. So Jonathan Peele has just, brought to the market a very nice book on the neuroscience of language, in which he's covering a lot of language and its neuroscience. And in the case for production, he's giving what he's saying, I'm giving the simplified model of what production entails. So there's lexical selection, what do I want to say, phonological encoding, what are the sounds involved, and then motor plans and execution of that motor plan or articulation. But when he's treating that in the way he says it, he already mentions there are definitely more complex models of it, but then the two models that he cites are, again, motor control models, right? So these models are preoccupied with how you articulate, but not so much with how do you select the words you want to say. And then he also presents the evidence of brain errors related to speech production. And if you see what they are actually errors related to motor planning, motor execution, kind of auditory cortex from you hearing yourself. But these are then more sensory motor regions than other things, right? And he's using the same term, speech production, as someone else that would be referring to something else. I also thought, let's see what Google, and these days Google gives you this additional AI thing that, you may like it or not, but you get it regardless. And I just put in there brain areas for language production. And immediately what you get from this AI overview is the primary brain area for language production is Broca's area, right? Which is located in kind of left frontal lobe, inferior frontal cortex. Also, the very next hit you get on Google is, again, Broca's area, is the area that allows us to articulate ideas as well as the use of words accurately in spoken and written language. So from the literature, if you would not maybe know very well this literature, it looks like Broca's area or sensory motor regions are the ultimate regions for speech production, right? And here also to give you another example of that, but now from kind of a scientific paper where the authors conducted a meta-analysis of voxel-based lesion mapping studies of aphasia. So these are studies that look at the relationship between areas that are lesioned and decrements in performance. And this is for lots of different tasks that are related to language impairment after brain damage. And then they grouped the tasks in terms of were people producing or were they comprehending language. And then they produced this statistical map of what regions are more likely related to an impairment in either production or comprehension. And what you're seeing there is basically for production, we are getting, again, a lot of this left inferior frontal cortex and lots of these kind of sensory motor areas, dorsal areas, and just a little bit of the temporal lobe there. So if you look across the literature, if you don't know exactly where to look, you get this idea that, Broca's area is it's a very important region for producing language. This long-standing interest in Broca's area, I think, is not only the case for people working on language, but also if you look at, comparative neuroscience, people working on evolution, they are also very much interested, or in the past at least, they were very much interested in Broca's area. And it could be that there's a kind of a relation there. The more we seem to think it's important for language, the more it may also be that during evolution, that was the area that enabled language to evolve. So if you look at some early papers, earlier, not so old, but still a bit earlier than these days, you find claims that, Broca's area expanded in size during evolution, possibly as an adaptation for our species' language abilities, right? So there seems to be, from the literature, this would be an extremely compelling idea that Broca's area is the speech production area, right? And then if you look at comparative neuroanatomy, it's also the potential precursor of a language-capable brain, because it's, the area that is, say, that lateralizes differently between humans and other non-human primates, etc. Right? So it's a very compelling story. Broca's area is the area for production. Production is unique to humans. Maybe what is unique in the brain as well is Broca's area. Except that some of the bits of this story are not exactly right. So one of the things, and I'm sorry, some of the things now on my screen apparently moved places, so I'm disturbed by it, you maybe not. So we conducted a meta-analysis, again, of voxel lesion symptom mapping studies of picture naming. And here we were focusing explicitly on picture naming as a way to operationalize language production. And then even more specifically, we were tapping into the retrieval from memory part of it, right? So we were not interested in, can a person really articulate or not? Is it at the motor level that they have a problem? But we were concerned with, can people retrieve a concept from memory? Can they retrieve a label for that concept from memory? The sounds that belong to that label, right? And then, so in the literature, you find these lesion symptom mapping studies, like I said, that map the relationship between tissue damage and task performance. And we conducted a meta-analysis of all of the available picture naming studies. So we included 10 articles in the end, testing 534 subjects in total. And here you find the results. So there were 2 main clusters. And this one in pink in anterior temporal lobe, and this one in blue that was in posterior temporal lobe. And it includes the inferior parietal lobule as well. Now, what I think you can immediately see is that there's no IFG in there, right? So Broca's area, which would have been here, I hope you can see my cursor. It's not there in our meta-analysis, right? So it doesn't seem to be a critical area for picture naming. What you can also see, right, and so if you would compare this to the results by Na et al. That found from, again, voxel lesion symptom mapping studies, a meta-analysis of those, that production is associated with left frontal cortex. Well, once you look at a narrower definition or and not in a negative way, narrower, right? But a better definition of what you're actually interested in, then you see that retrieval from memory for producing language is not associated with frontal cortex. It's rather the temporal lobe. This posterior cluster here that we found was also very much overlapping with the underlying white matter in there, right? So here in yellow, you see a mask of what the arcuate fasciculus would be, and in green, where our clusters overlap with the arcuate fasciculus. And so it seems to be the case that this posterior cluster is very much associated with the arcuate fasciculus. Now, that's we can't see your cursor. Okay, and I don't know if I can turn it on somehow. But yeah, I'll not try because I may break something. So I'll try to refer to the images more specifically. Now, we were doing all of this work on clinical populations, and on the other side of the building, literally, I had colleagues working on comparative neuroanatomy. And they were working with this idea of a connectivity blueprint. So the idea that you could describe cortical regions in terms of their unique sets of connections. And their point was that if we try to compare across brains of different animals, or different primates for that matter, it would be very hard if you're just, there are changes in size, there are changes in shape. It becomes very hard to know what is what in terms of the comparison. Is this region the same as that region? And so their claim was that if you would use these connectivity blueprints, you would have a common space for mapping these gray matter regions. You could then compare across species, right? So they were working on this idea. And I was so amazed to see what they showed to me at some point in discussions, that here they were making a comparison between the macaque and the human brain. And what you see in that color scale, the warmer the color, so say the more pinkish the colors you see in these brains, the more likely this area has changed in its connectional organization since the last common ancestor of humans and macaques. So basically, these areas that you see here are the ones that are most unique to humans, that you cannot simply explain by just some simple changes from the macaque brain evolving to the human brain. And I told my colleagues, like, this is amazing, because to me it looks very similar to what we are finding that are the critical areas for picture naming, right? So if you see here, again, I put the image that I showed you before from our own meta-analysis. You find this very nice, I find at least, that it resembles very much the areas that are unique to humans that cannot be explained by just some simple change in anatomy from the macaque brain. So we then, following this discussion, we sat down and said, look, we should be working together, because, you have knowledge of certain things and we have knowledge of other things. And once we put these things together, I think we can make a very interesting study about it, of regions that we know are important for language. Can we now tackle and look at whether they have changed or not in comparison to, or what their evolution is in comparison to the chimpanzee? So that's this study that we conducted with Joanna and Catherine as shared first authors. So one working on the human brain and the other one on the chimpanzee brain. And there's lots of different things in this study that I'm not going to cover, but I just wanted to show you these results. So here to the right, you see, if we seed from the posterior MTG, that is this thing, this part you see in yellow in the two brains. So the above one is the human and below is the chimpanzee. So you see from this yellow region, if we seed from there and we look at how the tracts are going, how the white matter pathways are going, you see already for the human, the upper one, that it seems to be making kind of a curve projecting dorsally, which you don't see in the chimpanzee brain. Right, so there's lots of other interesting things in the paper, but one of the findings is that the connectivity in the posterior temporal lobe via this arcuate fasciculus white matter tract expanded bilaterally to both frontal and parietal cortices in the humans, more so than in the chimpanzees, which very nicely aligned with our earlier findings from the VLSN studies. And more recently, there's also been additional data in that direction, a very interesting study that I thought would be relevant for you, if you know it already. But they also looked at this MTG region and how the arcuate fasciculus connectivity is coming from the temporal lobe, comparing chimpanzees and humans. And what you see here, so in blue, you see the superior temporal gyrus connectivity via the arcuate, and in red, you see the middle temporal gyrus connectivity. In the image to the left, and then in the image to the right, you see the comparison between chimpanzees and humans, the strength of the connectivity, of the connection. And what you see is that in chimpanzees, to the left in blue, the connectivity in the STG is stronger than it is from the MTG, which is in red. Whereas in humans, that's the opposite. So the connectivity through the MTG seed is stronger than it is from the STG, right? So you kind of get this reversed order between the two species. So they're saying that there is already a human-like arcuate MTG connection in the chimpanzee, but that is weaker than it is in humans. And they propose that this human-specific arcuate fasciculus morphology evolved by strengthening an existing connection rather than being something that arised, a new arrival. And they nicely say that this neuroscaffolding likely was already present in the last common ancestor and enabled the evolution of language processes in the human lineage, right? So now, rather than maybe being Broca's area, that is the potential precursor for language, it's rather we shifted a little bit towards the arcuate being a potential precursor. Now, this is all very nice, but something that I've been saying for a long time, to my lab and to colleagues, is that snapshots aren't very great for looking at language. So if you go back to the figure that I showed you before by Jonathan Peel, where we have an entire stream of the stages that you go through as you produce words. So first you have to select a word, then the sounds of that word, then the motor commands for that word, etc. It changes over time what is happening, and if you just have one snapshot of the entire thing, it could be very misguided. We could also rely on contrasts, right? So we have a task that tries to isolate one stage versus the other stage, but thinking that contrasts alone will always give us the right answer is, I think, asking too much from the operationalization stage, right? Because you need to be sure that you have absolutely isolated just that thing with your contrast, which I think, should accept that is not always the case, or often not the case. So what I've been working on a lot is on trying to add further converging evidence from a technique that does not only take a snapshot, but rather has temporal resolution, and that directly measures net neuronal activity, which is given to us by electrophysiology, right? So we are actually measuring what neurons are doing more so than if we have a snapshot. So I'm going to present data both looking at event-related responses, which is just time-locked and phase-locked activity to the stimulation that we are giving participants, or oscillatory activity, which is always present in the EEG. It's a bit less known than the event-related potentials in some fields, and it's also thought to reflect functional connections between and within neural networks. So I'm taking here to the right, you see, for us working on language production, the most prominent model of language production in terms of the cognitive stages that one undergoes when saying a word. And we're going to focus on the beginning of that model, because, like I said before, if we are giving people different tasks, we are in a way, tapping into various things differently because of our tasks, right? We have to go via tasks, but it also means that each task will have their own kind of artifact, if you will. So what is separated here in this figure to the right is this idea that you have a core process that you're trying to get at. So say conceptual preparation and lexical selection, but you come into that process via a lead-in process. So for picture naming, you have to recognize the picture. For, verb generation, you have to recognize the word you heard or the word you read. So these are kind of the leading processes, but they are not necessarily the core that we are interested in. So I'm going to present data from 3 different tasks, one of them being kind of the regular picture naming, so it's very much visually guided. The other one is contextually guided naming, so you will see an example of that later, but you get a sentence that gives you a context, and then you kind of complete that sentence via a picture naming. And verbal fluency, for which you are generating words and there's absolutely no visual input present. So if we start with contextually guided naming, the way this task works is that people have to name a picture, but before you give them this picture, they hear or read a sentence that is unfinished. So you get a sentence like, the farmer milked the, and then you get a picture of a cow and you name it cow, or the child drew a, and that is very unconstrained, right? Anything could complete the sentence. But the farmer milked the, usually there's a lot of priming through the semantic network that you get only 1 or 2 representations that would complete this sentence. So this is also in agreement what we find in behavioral data. So if you look at how long it takes people to name these pictures, in green, you see the constrained sentences, so the kinds of the farmer milked the cow. And in purple, you see the unconstrained ones, so the neutral sentences. And you see these lines that go like that. That's the entire reaction time distribution. So you have the 20% fastest responses at the bottom, all the way to cumulative to the slowest responses. And what you see is that there is a shift of the entire distribution to the left. So you're always faster if you're given context, right? So this really indicates that there's semantic priming going on. You are already retrieving pre-activating representations even before the picture is seen for the constrained sentences. If we look in the electrophysiology, what happens here, prior to people seeing this picture, it's a very consistent effect. It replicates over studies. We find alpha beta power decreases during this retrieval stage, right? So the way to see this plots here that I have above, you have the topographies, so how that effect looks over the scalp with EEG in this case. And you see that it has a left biased scalp topography. And then in these boxes below, you have time in the x-axis and frequency in the y-axis. And in color, you see changes in power, right? So here you get power decreases for constrained relative to unconstrained. That's what you see in blue, that precede the picture appearing. So before they see the picture, power in the alpha beta band goes down. We can source localize these effects using MEG. So here we had a study that brought people in twice to be tested just to see what is consistent across sessions. And in there we find that these alpha beta power decreases are consistent across sessions, so that's great. And they have this left posterior temporal inferior parietal lobule focus, right? So these are the areas that over consistent testing are the sources for these alpha beta power decreases. And this again, if you would compare that to the previous findings, looks again very much like the areas we identified in the other studies. Something else that I have the reference here, if you're interested in, if you have a lesion in these areas, you get rid of the behavioral effect. So you're no longer much faster in naming cow after a constrained sentence than after an unconstrained sentence. But you also obliterate the electrophysiological effect, right? When the areas lesioned are these ones. But if you lesion the Broca's area, it does nothing to this effect whatsoever. So it confirms that these sources are these posterior sources. Yeah, and that's what I mentioned before, right? So I think it nicely, again, resembles what we found with the meta-analysis of the picture naming via voxel lesion and symptom mapping studies. Now, if we go back to this idea of the lead-in and the core process, right? So usually in picture naming, what we get, that's the upper part of this schematic to the right, is that you get picture naming, you do visual recognition of that object, and that allows you access to the concept that you identify and you're going to name. And this conceptual preparation is a core process, but the object recognition is not, right? So in our unconstrained condition, the child drew, there's nothing in that sentence that is helping you. So in a way, the unconstrained sentences are just like picture naming, regular picture naming, right? You have to do it via the recognition, the visual recognition of that object. Whereas in the constrained sentences, what you get is priming through sentence comprehension. So you understand that sentence, there's, priming, there's some spreading activation through the semantic network, and that will lead you to also a conceptual preparation stage. But the leading process to that conceptual preparation is different, right? So now we can look at the core processes of retrieval, regardless of whether you came in via the visual system or through spreading activation in the semantic network. And that's what Natasha in the lab did. So she had people doing this task while she was running, this was an fMRI study. And what she did, and this one is a bit more complex to explain if you can't see my cursor, so I'll try to do this. So if we compare at this pre-picture interval, so that's before they see the picture, we assume, that's where you see the red arrows, we assume that that's when they retrieve already some representations of the picture that will be shown in the constrained condition, but not so in the unconstrained condition, right? Whereas if you look at the picture, the moment when you see the picture, in the unconstrained condition, you are retrieving it, but then guided via the visual system. And you're no longer retrieving it for the constrained condition because you already did it in the past, right? So these red arrows that you see are the assumed stages of retrieval that we are interested in. So you could say that if we look at the pre-picture interval, we are looking at contextually guided naming. And if we look at the picture interval, we are looking at visually guided naming, okay? And you have those here above. So you see the box for contextually guided naming, which, there's some, again, there are differences because this is a different technique, but there's also areas overlapping with our previous MEG work that you see there in blue. And there's visually guided naming, but what we were interested in is what is the overlap between these two areas, between these two contrasts, right? Because that would give us the core retrieval areas, whether it is via one way or the other that you get into, conceptual preparation and lexical selection. And those are the areas that you see in purple, and I guess it's hard for you to see in the figure exactly, but I've listed them here to the left. So these core retrieval processes are associated with bold increases in left fusiform gyrus, the mid portion of the middle temporal gyrus on the left, and left inferior frontal gyrus, which would be, the famous Broca's area, which you find here with fMRI, but you don't find in the MEG task, when we run this task with MEG. But we see this middle temporal gyrus so temporal lobe areas that are overlapping between these two ways of retrieving the concept and their labels. We're also very fortunate to work with a patient who had, I think, in terms of the lesion, a very, very big lesion. It's terrible, the size of her stroke. But it aligned very nicely with the lesion where it was with the work that we were doing before. So she had a stroke at a young age, and she had chronic aphasia. And what you see is in the upper row, you see the structural MRI, and in the bottom row, you see the tractography. And what you see in the three images to the left is in yellow is what would have been the arcuate fasciculus, right? So her arcuate fasciculus on the left is severely damaged, but it's intact there on the right. Now, she did these these 2 tasks with these 3 conditions that I showed you before. So she either named pictures following constrained sentences or unconstrained sentences, or just bare pictures, right? Which would be just, again, your typical picture naming study. And first, what we saw is that if you look at her word finding problems, so that the times when she could not name the picture, these are in this graph, the 3 bars to the right. So those are the word finding trials, the trials in which she told us, I know what I want to say, but I don't know the word for it. And you see that in light blue, when the sentences are constraining, she is much more likely to be able to retrieve those words. So the blue bars over the right for word finding are much smaller for a constrained condition. Whereas if we don't give her any context, that would be the darker blue bar. Or if she's doing bare picture naming, that would be the green bar. They are similar to each other, and they are about 50%, right? So about half of the time she can name the pictures if you don't help her with any kind of semantic cues. When we looked at her neurophysiological responses, so here in the upper row, you have a matched control that was matched in age, education, and sex. And then below you see the patient. And this is again the source reconstruction of the alpha beta power decreases in this pre-picture interval. And this is now a single subject, right? So it's different from the figures I showed you before because those were at the group level, but still for the matched control, you find this source in the temporal lobe and all of these more sensory motor types of regions, but it's strongly left lateralized. And then in the patient, what you see is that it's strongly right lateralized, right? But what I think is really exciting about these results is that what is right lateralized in her case is pretty much the same type of physiological response that the control shows over the left, right? It's not that the brain is doing something in a completely different way. Neurophysiologically, it's the same response, it's just shifted hemispheres, basically. If we then look at her visually guided naming and we compare the cases when she can name the picture, right? So those would be when she 50%, about 50, well, 46% of the time, versus the cases when she could not. She had a word finding problem. It was 42% of the time. If we compare the event-related responses for these 2 types of trials, what we see is that she has an N400-like type of response. It doesn't really matter so much, but it basically means there's a peak around 400 milliseconds after she sees the picture. And this peak is there both for successful naming and anomia cases, right? So timing-wise, these responses are also similar, but there's a difference in how strong they are and also where they are generated. So if we look at the trials, the brain areas there have a stronger response when she can name it, so when she's successful in naming. These are the areas in red, and that's in the mid-posterior temporal lobe. And if you reverse this and you look at what areas are more active when she cannot name, so she fails to name it, those are the blue areas, and that's the right inferior frontal gyrus, right? So when she can name it, it seems, this is kind of a matter of a difference in strength between the 2, but if she can name it, the responses are stronger in right posterior temporal lobe, and if she cannot, they are stronger in right inferior frontal gyrus. So kind of this is just a plug-in and aside here. We are trying to see if we can study language in a more integrated way, right? Rather than, again, just there's a picture naming task and that's it. We proposed what we call in the CLAP, a concise language paradigm, that would really present a framework for studying comprehension and production together, right? And it makes use of these contrasts that I was just showing you. So you get constrained sentences, unconstrained sentences, reversed sentences, reverse pictures. And you can, in a way, have all kinds of low-level and high-level contrasts in one paradigm. So the task for the participant is always the same. So if you're interested in that, you can look at that reference. So this was all, driving the system via, either the visual input or via the sentence that was presented to the participants. And then we can have yet another type of lead-in, a task leading to another kind of leading process, which is, generating words, right? So we would, I don't know, do verb generation or noun generation. So you get the letter F and you have to produce as many words as you can, starting with the letter F, for example, right? So that's the verbal fluency task. Or I say animals and you have to give me as many animals as you can. And so in here, you also get into the core processes that we are interested in, these core retrieval processes, but the way to get to them is again via a different route, right? So the lead-in is different, but the core is the same. And now the question is, do we find converging evidence there as well? And this is work we've done using intracranial EEG data, so that's also why I thought it could be relevant for you, for this audience. So we had patients with left hemisphere coverage, and they did this task in blocks. So they had blocks of categories, blocks of letters, and blocks of rest. And we are contrasting the blocks. We're always contrasting a fluency block with a rest block. And we are interested in the time window between 750 and 250 milliseconds before speech onset, right? This is kind of the issue with the verbal fluency task. You don't have a stimulus that drives the response, so the time locking is a bit trickier. So we take a time point for which we know that it should be about the retrieval, more or less. And then we are using a Bayesian analysis type of measure that gives you the prevalence estimate in the population of that effect. And what we find when we look at retrieval stages, the retrieval period for producing these words in the verbal fluency task is, again, we find alpha power decreases in the left temporal lobe, right, which you see here in blue. And there's also inferior parietal. But again, it's alpha power decreasing in the temporal lobe, in middle temporal and inferior temporal gyri, similar to what we found with the other areas. So to me, that's nice converging evidence across different tasks. So trying to summarize it for you, if we go task by task, we see that, from lots of research out there, not only I presented you just a little bit of that, but there's a lot out there showing that visually guided naming, when we look at these, stages of really retrieval from memory, more so than just a motor command, then we find a critical role of left temporal lobe and inferior parietal cortices for that. When you look at contextually guided naming, which would be, in a way, different task, yet we are tapping into core processes of word production, we find that they are reflected in alpha beta power decreases, again, in left posterior temporal and inferior parietal cortex. And there's some causal evidence in there that I showed you very briefly. When we look at noun generation from verbal fluency, again, during the retrieval stage, we find alpha power decreases in the temporal lobe, right? And so there's partial overlap across all tasks, of course, because we are always dealing with this, what is lead-in and what is core. But ultimately, there seems to be some convergence across word production tasks, tapping into this word retrieval for speaking. And that is, to me, interested in neurophysiology, I think that's beautiful that this convergence is not only spatial, being in the posterior temporal lobe, a lot of MTG and also ITG or fusiform gyres, but there's also convergence in time and in frequency, right? So across various tasks, when it is possible to do that analysis, you find alpha power or alpha beta power decreases. There's also convergence in terms of left and right hemisphere homolog areas, right? So the neurophysiological phenomena that we usually measure on the left, we also measure them on the right in these homolog areas. So there's convergence also in terms neurophysiological responses themselves. And in a way, maybe that's also not so surprising, right? So if you go back to these quotes from these papers, the connectivity in posterior temporal lobe expanded bilaterally to frontal and parietal cortices in humans compared to chimpanzees. So indeed, if there's a scaffolding that is there on the left that has a certain neurophysiological response, it should maybe also be there on the right in a similar way, because the scaffolding should be similar. So I think that there's a lot that can be looked at, in my opinion at least, in terms of comparative neuroanatomy, not only at the level of is it this area or not, is this area on or not, on or off, when we look at snapshots kinds of things in trying to make comparison across tasks, but also in the types of neurophysiological responses that some of these areas produce. So with that, I want to say, if you forget everything that I told you, then at least remember this thing, that the production of language depends heavily on the temporal lobe structures. So not doing ba 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 ba, because that's to me not language, but once we look at the production really of content in language, that really depends on temporal lobe structures, way more so than on frontal cortex. So thanks very much for your attention. There are some people that would like to thank specially for all of their collaborations over the years that allowed me to do all of this work. And I also have here a plug-in to my efforts in putting together, with the help of lots of other people working on language production, a resource on language production, but that is for free, written by experts on language production, that I think if you Google this, you will find an access to the whole book. And that is really to, make justice to language production as such, rather than being the forgotten part of language. Thanks very much, and I'm happy to take questions.
Thank you, Victoria, for the amazing talk. Now we have some questions in the YouTube chat. The first one is there. Could using mental imagery prior to picture presentation in a picture task facilitate lexical retrieval in individuals with aphasia?
That's not exactly my expertise. I think if it's just imagery of the concept of a kind of just an object itself, it will not help, because the issue that they have is really with, say, the word that goes with that concept, or the sounds that form that word that goes with that concept. So I think just imagery of an object itself would not help, also because they know, if they can't say, I don't know, cat, but you show them the image of a cat and an image of a dog, and you say, is it this one or this one that you mean? They will know that they mean the cat, but it's really the word cat that doesn't get to them. So I don't think it would actually help so much, the mental imagery as such. Yeah.
Okay, thank you. Next one. Are these responses or signals related to age?
Yeah. So we've been looking a lot into it. I think the answer is it's complex, but there's some work showing that in general there are changes to the frequencies get, I think, slower as people age. So there is a general change in there, and also that, at least in our work, it seems to be the case that as people age, these responses become more bilateral than when people are younger. So there's also a change, not so much to the responses themselves, but to how much the right hemisphere is contributing in generating them. So yes, there's a lot of interesting work to be done there for sure. Yeah.
Amazing. Okay, next one. Yeah, I'm seeing. Guys, will you put on the screen? Okay, thank you. How does understanding language as a network can contribute to assessment and rehabilitation of patients with aphasia, especially those with temporal lobe lesions?
Yeah. I started doing work on this a very, very long time ago. And I will be very honest with you. I had this idea that if we understand the brain, we'll be better able to, also have better assessments, better diagnosis, better rehabilitation plans, etc. And I've changed my mind over the years because I find that the gap between behavior and the brain is just so vast that if we want to improve something in behavior, we need to tackle behavior. If you want to do better assessments, do better behavioral assessments. And knowing more about the brain, maybe it will help you in terms of saying the temporal lobe is lesioned, therefore, most likely this person will not reach that particular level. But then very often we are surprised again because the brain will do things that we didn't expect it to do. So I think in the end, if you really want to make a difference, then behavior should be understood better than we do now, more so than understanding the brain better. But that's me getting old and grumpy and thinking that it's just the gap is just too big.
That yeah, a really nice point of view.
Sorry to disappoint many of you.
Okay, Leonidas, are there any essential analytical techniques that support the involvement of brain areas beyond the frontal cortex in language production?
I'm not sure I get the question. If so, analytical techniques in order to study the involvement of brain areas, is that what you meant?
I
I don't
guess
know if the question can be clarified.
I think it's if there are some of these techniques that are crucial to this type of study, I guess.
Okay, yeah. If you're interested in, let's say, in how things are changing even over the time of a second that it takes, less than a second that it takes you to produce a word, then you really need temporal resolution, and then you have to work with electrophysiology. Right? If you're more interested in really just the spatial dimension and you want to know is it this area or that area, then I think you would go for a technique that gives you snapshots with great spatial resolution at the cost of only having a snapshot. So something like fMRI would be more advised. And if it's really about is this area really critical or not, then you need lesion models for answering those types of questions. But I'm not sure if that's really what the question was trying to get at.
Ok, thank you. Bom, qual a expectativa a respeito da evolução dessas áreas em outros vertebrados além de primatas? I'll translate to English if there's any people from abroad. What's the expectation related to the evolution of these areas in other vertebrate groups beyond primates?
And now I must say I'm completely out of my comfort zone. I would think that Erika maybe has more to say about this than I do. No, but I don't think many other animals even have a temporal lobe, a developed temporal lobe, right?
Yeah, we have some different things. Some different things are similar in different vertebrates, but I think we're really far from this level of understanding. I don't know. I'm still starting this world.
Yeah.
But yeah. Okay, now I have one question of myself that I put on the chat. Human brains are more connected than chimpanzee brains, and this underlies our capacity of language, right? Thus, is the loss of this connectivity in damaged human brains the main cause of this speech impairment? Can we make this type of generalization already, or no?
Yeah. I think what we learned from the study that we did together is that actually it was not the case that the human brain was more connected than the chimpanzee brain, because there were also types of connections that were much stronger in the chimpanzee brain than they were in the human brain. So that was to us very interesting. It's not everything in the human brain got more connected, which is actually also really cool, right? So the answer was not everything is better in the human brain, more connected, more complex, more. That was not the case. In certain, I think these were ventral, so kind of via the temporal lobe, these were stronger in the chimpanzee than they were kind of just within the temporal lobe itself than they were in humans. I may be misremembering this, but I think that that was the finding. So it's not exactly the same. It's not exactly so that it's more connected overall, but for the arcuate fasciculus in particular, yes, that
Mm,
was stronger in humans. And it's also what this other study found later, right? That chimpanzees have it, but it's just much weaker. And these are the areas that I think people working on brain damage are more and more confident about saying, if those are damaged, then you will have a language impairment. And that's why for us it was so beautiful to do this collaboration, because we had this knowledge of, if you damage this area, you get a language impairment. And now you're telling me that this is the connection that is unique to humans. And that was indeed, I think, being careful with making generalizations, but yes, it seems to be something of that kind. Yeah.
amazing.
Yeah.
Okay, Pachi, how can this more distributed view of language influence or benefit areas like brain-computer interfaces or speech-related artificial intelligence?
Yeah, that latter one I wouldn't really know, but I do think that it is distributed, but also maybe not, because I think it's distributed in terms of, again, if you take the snapshot kind of uh, metaphor, I don't even know if it's a metaphor, but if you look at language production as a whole from beginning to end, then it will look like it's distributed, right? Because you have to go all the way from, I don't know, from activating a concept they want to say to the words with it, to the sounds with it, to the motor commands with it, and then it looks like it's really distributed. But if you're focusing only on, I don't know, articulation, then it's not distributed. It's very much just a motor cortex kind of thing. So I think it's not even so much the knowledge that it is distributed that would help, but understanding that it is something that changes, that has to go from the one location to the other in time, and acknowledging that there's a time course to it, and snapshots won't always give us all the answers, that is actually an improvement rather than saying it is distributed. I think it would help these fields if the questions are more targeted at a particular
Okay.
stage, if you will, or is it more retrieval from something from declarative memory, or is it more a motor thing that I'm interested in? I think that that's what is lacking.
Nice. Okay. Why do you think the idea that temporal lobe is solely responsible for comprehension and the frontal lobe for the production still persists, despite so much evidence to the
I don't
contrary?
know. I really don't know what happens. And sometimes I'm talking to neurologists that I collaborate with, and they tell me like, wow, this is new to me. That's not what we have in our textbooks. So I don't know if it's more a knowledge amongst us working in that field. But if I go to someone who actually does not work on, doesn't have much knowledge of brain damage and language impairment following brain damage, then I think they would still ascribe to that kind of view, more because these subfields are not necessarily talking to each other so much.
Yeah,
Maybe
yeah,
it's a little bit of that. Yeah.
there are another comment. Oh, okay. After, if you could, no, I just complement with the comment. Okay, go ahead. If you could test the ideal patient or several patients in an intracranial recording setting, but had a limited time, which one to tasks would you choose to administer? Which
Uh,
one?
yeah, I would say first of all, uh, it would depend a lot on what the question is that I would like to answer. But I'm going to, I'm biased, but I would say our clap is really meant for that, the concise language paradigm, because it gives you a low level sentence comprehension contrast, a high level contrast, a low level object recognition contrast, a high level contrast. So you can do one task and you will have picture name, you will have sentence comprehension, you have this context effect. You can, if you call it prediction, you will have prediction. You can play with low and high frequency where it's all in one task. So I would do then only one task, but then this one.
Okay. Just a comment on the previous question was on the, yeah, this one. That it's a problem, right? Because
Yeah,
you have
exactly.
students of medical students learning
Yeah,
old concepts that are not
yeah.
right
Yeah,
anymore.
right. Yeah, exactly. And this is, I think, not only in Brazil, because these colleagues I talked to, they are in the Netherlands, right? And they say this is what the textbooks tell us. Yeah, yeah. So
Okay. Now, okay, yeah. So great talk and some compliments.
thank you. Yeah, and Carlos, yes, I think this is very true. I'm working a lot with a philosopher now on this issue. I think we really need better conceptual clarity in what we are doing, for sure. Yeah.
Okay, now I think we already passed our 15 minutes. So thank you again, Victoria. It was an amazing talk. We learned a lot. Thank you to having to be here. And everyone, pay attention that next Thursday we have our last webinars. And that's it. Thank you for your attention.
Muito obrigada pela oportunidade.
Eu
Tchau.
que agradeço. Tchau, tchau.
Tchau.